We'll begin the webcast with a series of presentations from our guest speakers. Our first speaker is Dr. Christoph Zimmer, Research Director at the Institut Pasteur in Paris, France. Our next speaker is Dr. Florian Jug, Research Group Leader, Center for Systems Biology, Dresden, the Max Planck Institute of Molecular Cell Biology and Genetics, Dresden, and Fondazione Umane Tecnopole, Milan, Italy. Our final speaker is Dr. Martin Weigart, Group Leader, EPFL Lausanne, Switzerland. We'll then move on to a question and answer session with all the speakers. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here and then press submit. Since we have many speakers today, please do indicate to whom you would like to direct your question, and we will do our best to answer them live today. And now, over to Dr. Christoph Zimmer.
Welcome to this webinar. So my plan here is to give a short introduction to deep learning in just a few minutes, and then to highlight some of our work on applications of deep learning to super resolution microscopy. So let me start with the first part. So deep learning is a subset of machine learning, which itself is a subset of artificial intelligence. But deep learning is probably the reason why there's so much talk about artificial intelligence in recent years. And deep learning has been around for a long time, actually, and uses methods known as artificial neural networks, and more specifically deep neural networks, that can learn very complex relationships between inputs and output data in the most standard use. So the reason that these methods are called neural networks is based on the loose W2, X3 multiplied by W3 are added together. And on top of that, we add another number, which is here called W0, which is also called a bias. So this computation is very simple. This is just a linear combination with an added constant number. And this is called a pre-activation. And this number is then passed through another function that has to be nonlinear. So there are multiple functions that can be used. They're called it's called f here. And the more standard function that is often used is called the ReLU function, which is simply a function that takes a maximum between 0 and x. So if x is negative, the output will be 0, and if x is positive, the output will just be x. And this nonlinear function is a critical component of neural networks. And so the output, so f of z, will be sent to other neurons downstream. Okay, and so the weights and the bias, so W0 to W3, are the parameters that are associated to this single neuron. And so if the weight, for example, W1 is positive, you can think of the neuron X1 to have an excitatory effect on the central neuron. If this weight is negative, it will essentially have an inhibitory effect. And so you can think of the connections between neurons as synapses here. And the critical aspect of deep learning is that these parameters are going to be learned. And we'll come back to that in a few slides. So as I mentioned, these neurons are connected to each other in larger networks. So on the left, we're looking at a very simple neural network that consists of an input layer that will just represent the input data. So in this case, just 3 numbers, x1, x2, x3. And then we have 2 layers of 4 neurons each, and we have an output layer that in this case consists of a single neuron, but there could be many more neurons. So we have here 2 so-called hidden layers, a total of 3 layers. And if we now you count the number of arrows between the neurons, you will get the number of weights. And if you add the biases to that, you will see that we have here 41 parameters in this simple neural network. But in standard or state-of-the-art applications of deep learning, we now use much bigger neural networks, such as, for example, the ResNet 152 neural network that has an architecture composed of more than 150 layers, consecutive layers of neurons, connected by so-called skip connections. And the total number of parameters here exceeds 23 million. And because the neural network has so many parameters, it is actually able to learn very complex transformations between input and output data. So just to highlight what you can do with these neural networks, and also to explain the basic principle of learning, I'm using the classical textbook example of image classification or image recognition. So the task here is simply to take an image as an input and then to give an output that will correspond to an object, or in this case, an animal contained in the image. So this is a classification task where the input is an image and the output is a category, let's say a word among a list of predefined words, cats, dogs, etc. And so the task here of image classification is to be able to, for the computer to analyze this matrix, this array of numbers, in such a way as to determine that this actually contains a cat. Okay, so this slide now tries to explain the basic principle of learning in a neural network in the context of image classification. Here in this textbook example, we simply want to train a neural network to distinguish images of cats from images of dogs. So the way we are doing this is that we are providing the neural network with a set of training data. These training data are images of cats and dogs that have already been annotated by a human, which means that they are being given labels. So the images of cats are being given a label of 1, the images of dogs are being given a label of 0. And now we are training this neural network to be able to produce output numbers for catness and dogness. So the target numbers will be 1 for cat and 0 for dogs, which are in fact given by the labels. But at the beginning, when we are feeding these images through the neural network is going to compute very different numbers because the parameters in the neural network have been initialized randomly. So this neural network at the beginning has not been exposed to any data. And because these numbers have been initialized randomly, the output of the neural network will consist of numbers between 0 and 1. These numbers will have no relation with the actual content of the image. So, for example, we could get 0.4 for a cat or 0.3 for a dog, as shown here. So initially, the neural network is not trained, but because we know what the correct answer is for each of the image, we can compute an error. So, for example, here we are using the mean squared error. In practice, one would use something more elaborate called the cross entropy, and this defines the so-called loss. So at the beginning, the errors will be large. So, for example, 0.4 minus 1, this is 0.6 in absolute numbers, and this is going to contribute to a substantial error if you add this to all across all the training images. So this error is large, but we are now going to use an algorithm called backpropagation that can change very slightly the values of all the parameters in the neural network in such a way as to reduce this error slightly. This is illustrated here. And now you can see, if you compare to the previous slide, that the numbers are moving in the right direction. For example, for the cat on the top, we are going from 0.4 to 0.5, so closer to the target value of 1. And so this operation called backpropagation, which involves computation of lots of partial derivatives, is being done many times by after randomly shuffling the training images. And after many, many iterations, we are ending up in a situation where the predicted catness values are in fact very close to the labels. Okay. So this is the training stage. In itself, this would be of no interest whatsoever because the images have already been annotated by a human. So we know which image is a cat and which one is a dog. But the beauty of deep learning is that the neural network that has now been trained is able to generalize to new images of cats or dogs that have never been seen before, neither by a human nor by a machine. And one of the early breakthroughs achieved by deep learning is the classification of images in a much more complex scenario than the cats and dogs I talked before. To be more specific, a lab in Stanford had designed a challenge called the ImageNet Large-Scale Visual Recognition Challenge that has assembled a database of more than 1 million of images, which have been annotated in 1,000 or more different categories, as you can see here: container ship, motor scooter, leopard, etc. So a large variety of input images that have been annotated by humans, and then computer vision groups that participated in the challenge have used algorithms to classify these images, and they were allowed to provide 5 predictions, 5 categories for each of these images. So based on this definition of success and failure of image annotation, you can quantitatively compare algorithms that participated in the challenge. And in 2010, the best entry in this challenge made an error of a bit less than 30%. And by comparison, the human error, the average human error is 5%. So the computer vision algorithms had pretty low performance, pretty bad performance in 2010. And so the situation improved a little bit in 2011 with an incremental improvement of 2%. But then in 2012, the classification error of the best entry dropped dramatically by 10%. And they used deep learning to achieve this. And since 2012, there have been lots of improvements, very rapid improvements in this image classification challenge. And all of the winning entries are now using some form of deep learning. And now the performance is better than the average human performance. So this is one of the main breakthroughs that has essentially triggered current interest in deep learning and also artificial intelligence in general. Now, more importantly, perhaps, there are now applications of deep learning in real-world applications that go beyond the computer vision domain. For example, in medical imaging, here a team has developed neural network algorithms that are able to detect a particular medical condition called diabetic retinopathy just by analyzing these images of the eyes. And this deep learning-based technique has been shown to outperform the diagnostic accuracy of certified ophthalmologists. Another very different application is speech recognition. Essentially, now all smartphones use some form of deep learning to convert speech or voice recordings into text. Another very interesting application in structural biology is the use of deep learning to predict three-dimensional protein structures only from the amino acid sequence. And here, again, deep learning methods have been shown to outperform other structural biology methods in a competition. There are also applications for games where it has been shown that deep learning can now outperform any human in complex games such as Go. Okay, so despite all these successes, I want to stress that there are also important limitations of deep learning, including in the case of image classification that I mentioned before. For example, here is a deep learning method that has used an image as an input to produce an output sentence, and clearly the sentence that the boy is holding a baseball bat is not correct. So, and this is most likely an error that no human would have done. So deep learning models can be very accurate, actually more accurate than humans in many settings, but they can also fail in dramatic ways that in which humans would not. Another related and very important point is that one has to be very careful of so-called model mismatch. So in general, one has to be very careful with the application of deep learning to data that are of a distinct nature than the data that have been used to train the neural network in the first place. Okay, so as a summary of this very brief introduction, the basic principle of deep learning is that artificial neurons will take inputs from other neurons, will compute a linear combination of the values provided by these numbers, by these neurons, with some trainable parameters, then apply a nonlinear function and pass the output to other neurons, and so on. And these operations are going to cascade through a large neural network. And because these networks contain so many different layers and neurons and often millions of parameters, they are able to learn very complex transformations between input and output data. And fundamentally, neural networks need to be trained, so the parameters are learned iteratively using this algorithm known as backpropagation and also stochastic gradient descent that tries to minimize a loss function based on training data. in supervised learning, as I mentioned, the neural network is trained on labeled data, but there are also unsupervised forms of learning that I did not have time to mention. I also did not have time to mention the recurrent problem of overfitting, which has to be taken into account in any deep learning application, but there are many ways to diagnose overfitting and also to remedy it. Now, deep learning is very good at tasks that can be called as perceptual association and is at the heart of many breakthroughs in many applications, but it lacks a human level understanding of the content, for example, of image scenes, as illustrated by the example of the kid with the toothbrush, and can fail miserably in very unhuman ways, particularly in the case of model mismatch. Okay, so this is the first part in my introduction to deep learning. Now I would like to move on to an application that we developed where we are using deep learning to enhance super resolution microscopy. So as a very brief introduction to super resolution microscopy, it is well known that fluorescence microscopy is a key technique in the life sciences because it allows to look at cells with molecular sensitivity. So here, for example, in this movie, we can see tubulin in green and the Aurora B protein in red. But a key limitation for a long time has been the diffraction limit, which, as a result of which, objects smaller than half the wavelength of light cannot be resolved. So, for example, if we take an image with a conventional fluorescence microscope of a nuclear pore complex, which is a huge molecular machine but only measures about 100 nanometers in size, we are going to see a blurry pixelated image with a width, full width half maximum, of approximately 200 nanometers. And if we look at another completely different biological entity, such as an HIV particle, which also has a size smaller than the diffraction limit of roughly 100 nanometers, we will see essentially the same. So we will not be able to distinguish any structural details from these images. But since at least 15 years or so, there have been breakthroughs in the development of so-called super resolution microscopy techniques. And one particularly powerful approach is called single molecule localization microscopy, also known as PALM, STORM, or various other acronyms. The principle of this method is surprisingly simple. Instead of taking a single image where all molecules fluoresce at the same time, you are taking a sequence of images in which the fluorescent molecules are allowed to blink. And they are allowed to blink in such a way that individual frames will contain only a few fluorescent or active fluorescent molecules. And because these molecules are sparse, because they are spatially isolated from each other by distances more than the diffraction limit, they can be localized with high precision using adequate algorithms that are well known in the single molecule field. And from these localizations, one can generate an image in the computer that in fact represents a high resolution view of the structure. So in order to do this, you need to be able to obtain localizations from many molecules. And because we can only allow a small number of molecules to be active in each frame, this means that the number of images of diffraction limited frames that needs to be acquired in order to get a good super resolution image is very large, for typically tens of thousands or even more, sometimes hundreds of thousands of frames. So there is so these methods allow a very substantial improvement in the resolution, sometimes by a factor of tenfold, sometimes even more. but the temporal resolution is made worse by an even larger factor of 10,000 or even 100,000. So these techniques are extremely slow, and it is often considered that there is an intrinsic trade-off between the spatial resolution and the temporal resolution, as illustrated by this little pyramid here, and that one can only improve the spatial resolution at the expense of the speed, or vice versa. But so here in this project, we try to address this situation using deep learning. So we developed a method called artificial neural network accelerated localization microscopy, or in short, ANAPAM. And when I say we, I should say way, because this is very largely the work of a former PhD student of mine called Wei Ouyang, with help from Andrea Ristov, Mikhail Lelek, and Shen Hao for the experimental part. So the basic principle of that method is as follows. So we are going to train a neural network to produce output images from input images. And in this case, we are going to use traditional SMLM imaging to define the training data. So we are going to take typically tens of thousands of low-resolution images, like I mentioned before. And from this, we can generate, as I mentioned before, using standard localization algorithms, high-quality super-resolution image called here PORM with capital K. But we can also take just a subset of the raw frames, so let's say just a few hundred instead of tens of thousands, and from these first few hundred of low-resolution images, using the same localization algorithms, we can obtain what we call here a sparse localization image. So call here P small cap K. And at the beginning of the image sequence, we can also obtain a wide field image of the same field of view. So what we are now doing is that we are going to train a neural network here called ANN to reconstruct an output image called here AK that should be a good approximation of P capital K, of the image that we would have obtained with many more input frames, with tens of thousands of frames. So we're essentially trying to, in some sense, predict the localizations of molecules that have not been yet imaged. And in practice, the way we do this is that we define an objective function, which is another word for this loss function that I mentioned earlier, that measures the quality of the reconstructed or output image AK in comparison with PK. And we ended up with a relatively complex loss function that contains three terms. I don't have time to go into the details, but the first term here essentially measures the agreement between the AK and P capital K, so between the ground truth image and the output image, using L1 norms and multi-scale structural similarity indexes, which are established ways to compare image similarity. And on the bottom, we use another device called a conditional generative adversarial network error that is able to determine if the output image is consistent with the probability density that has been seen in training data. And we also have a third term, which we call the low resolution reconstruction error on the center right, which essentially measures the agreement of the reconstructed image AK with the low resolution input image W or L here. Once the neural network has been trained, it can be applied to new images, to test images. This is also called inference. So here we are taking another image on another day but of a similar structure, like in this case, this cartoon symbolizes microtubules. And instead of taking many thousands of low-resolution images, we take just a few hundreds, and also a wide field image, and we use these two inputs, so the low-resolution image and the sparse localization image, as input to the trained neural network. And the output on the right here is a reconstructed super resolution image that hopefully is in good agreement with the ground truth. And on top of that, we can also generate an error map that I will come back to at the very end. So this here is just the concept so far. Now let me show you some real data. So here on the top left, you are looking at the super resolution D-storm image of microtubules obtained with 60,000 input frames in roughly 10 minutes. And on the bottom, you are looking at a sparse localization image of the same field obtained from only 300 frames. So if you zoom at it, you can see that it's the images on the bottom is of very low quality and contains and makes it hard to distinguish individual microtubules, for example. But essentially, we are now training a neural network to try and reconstruct an approximation of the ground truth image on the top left from the sparse input image on the bottom left, with the wide field image as additional input, which I didn't show here. So in practice, we train this neural network on just a few fields of view. Here, 6 in total, shown on the right. And now the question is whether this trained neural network will actually work on test data, so data that have not been part of the training data set. And so this slide shows that it does. So you can see on the left the input data, so a wide field image on the top and a sparse localization image on the bottom. And then you can see the output of the neural network. And to the right, the comparison with the ground truth, which is simply defined as the super resolution image obtained in the standard way by taking many more frames. So in this case, 30,000 low resolution frames instead of 300 frames for the sparse image. And on the very right, with the superposition of this merged image that superposes the anapalm reconstruction, so the output, with the ground truth in 2 different colors, you can see that the two agree with each other very well, even though the agreement is not perfect. But essentially, all the structural details here that are important for interpretation of this image are faithfully recovered. Essentially, here we are achieving a reduction of image acquisition time by a factor 100. So what can we do with this? Well, since we can now accelerate the imaging, we can also look at many more fields. So traditionally in SMLM imaging, the imaging is restricted to a few tens of cells or a few tens of fields of view. Here we decided to take images from more than 1,000 fields of view that are consecutive to each other, so that we can essentially tile an area of roughly 2 by 2 millimeters. And the imaging here was done in a few hours, so in a reasonable time frame. And so by applying anapalm to these more than 1,000 fields of view, we can in fact reconstruct an image that covers spatial scales from approximately 20 nanometers all the way up to 2 millimeters. So we were ending up here with a very large image containing a bit more than 1,000 cells. So essentially, this is a method that can help reconcile the conflicting aims of achieving high resolution and high throughput imaging in our view. Now, I showed here results based on microtubules, but the technique is not restricted to that. It's much more generic. So for example, we can train a neural network on images of nuclear pores. So here we use the exact same architecture, same hyperparameters, etc. But we retrained a neural network on images of nuclear pores here using immunolabeling of the nucleoporine GP210. And what you can see on the top left is the input sparse palm image, and obtained from 3,000 consecutive image frames. In the on the top center, you can see the output of the neural network, which clearly shows multiple ring-like structures as expected for nuclear pores in super resolution. On the top right, you can see the ground truth image obtained from 10 times more frames, from 30,000 frames. And here we argue that the quality of the reconstructed image is in fact higher than that of the ground truth, because you can see some complete rings in the reconstructed image that you cannot see even in the ground truth. So this, of course, raises the question as to whether these structures are real. And in order to verify this, we use another dual color imaging of a lectin, WGA, shown in green, that is known to bind to the inner core of the nuclear pore. So here we did not process these images of WGA with ANAPALM. We simply overlaid the super resolution WGA images to the ANAPALM reconstruction. And you can see that the rings that have been produced in the output of ANAPALM are in fact verified because they have a green dot in the center. But we also applied ANAPALM to mitochondria, which are much more complex structures, as you can appreciate here. And again, these images here show that ANAPALM is in fact able to reconstruct the morphology of mitochondria. However, if you look in more detail, you can also see discrepancies. So the distribution of proteins along the membranes is less well reproduced than for nuclear pores or mitochondria. This is not surprising because these structures are more complex, and we did not train the neural network on more data. We retrained the neural network here on mitochondria images, but only up to 10 different fields of view. Here we have been able to reconstruct a super resolution image that has a quality similar to what you can obtain traditionally, but taking much more images, even though we did not change anything to the image acquisition protocol. So in some sense, we are compensating the lack of information from in the input data using prior information that has been learned previously by the neural network based on the training images. And this raises a number of questions. In particular, it raises the possibility that the outputs of the neural network might be biased towards the training data. And the related question is whether this method can actually allow to visualize changes of the testing data relative to the training data. In other words, that does it enable to discover features that have not been seen before, or in other words, how robust is this method? So so these are very important questions. And in order to try and address them, at least in part, we run two different experiments. So the first experiment is that we trained a neural network on microtubules and then applied it to analyze images of nuclear pores, which are obviously very different structures, as you have seen. Now, here is on the left is again a sparse image of nuclear pores, and on the top right is the output of a neural network that has been trained on nuclear pores, on GP210 proteins. And in this case, the reconstruction, as I showed before, is in good agreement with the ground truth, is of good quality. However, if we now use the same input image but feed it through a neural network that has been trained on images of microtubules, then the output is very different. It contains apparent filamentary structures that have no resemblance with the ground truth whatsoever. So this is a very strong artifact here. And so this illustrates that this technique, as essentially all related deep learning techniques, can generate hallucinations if used improperly. However, it is unlikely that anyone would use ANAPARM in such a way. So a method trained on microtubules should not be applied to nuclear pores. A more meaningful question is whether a neural network trained on microtubules in one condition can be used to analyze microtubules in other conditions. So we decided to test this, and for this we first considered images of microtubules in absence of any drug treatment or perturbation. So these are the same kind of images I showed before. We have here control cells. We have an input sparse input image. We have an output from ANAPARM that is in good agreement with the ground truth. This is exactly what I've shown before. But now we are going to treat the cells with Taxol, which is a drug, an anti-cancer drug that tends to make microtubules more rigid and stiffer and parallel to each other. So it induces a change in the morphology of the microtubular network. And we did not retrain the neural network under this new condition. We used a network that was trained previously on untreated cells. And here again, the output is in fact in good agreement with the ground truth, as you can see here. Now here the perturbation was perhaps relatively subtle, so we tried a more massive perturbation using nocodazole, which is a drug that depolymerizes most microtubules. And as you can see on the bottom left sparse input image, there's much less signal because most of the microtubules here have been destroyed, but the output of ANAPARM is still in good agreement with the ground truth that is obtained here with 60,000 frames. So despite this strong perturbation of the structure by the nocodazole drug, the network has been able to reconstruct a good approximation of the ground truth, suggesting that it is indeed robust to these kind of perturbations. Okay, so here we have shown that ANAPARM is essentially robust to chemically induced changes to the microtubular network. But the more general question remains whether we can apply this technique to different types of conditions or data that ANAPARM has not been trained on. This is a fundamental question in deep learning in general, but here we are providing at least a partial answer in the very specific context of ANAPARM, where we are comparing the consistency of the output of ANAPARM with the input wide field image. I briefly mentioned this arrow map in earlier slides. So what you can see on the right here is a map that highlights the discrepancies between the predicted or reconstructed ANAPARM image and the input wide field image in simulated images of nuclear pores. So what you can see here is that the error map lights up, becomes red, when the simulated size of the nuclear pores becomes very different from the nuclear pores that have been used in the training data. And this method, which is directly inspired from work from Ricardo Enriquez' lab, is able to provide a warning that there might be something wrong in the application here of the neural network to these data, essentially because there is a model mismatch. Okay, so in summary, I hope I've convinced you that this technique, ANAPALM, can accelerate super resolution imaging, essentially by predicting images from much fewer input information than are traditionally required. And in that sense, this approach is in fact able to overcome some of the apparently fundamental trade-offs between spatial resolution and temporal resolution and speed. And I've shown you that ANAPALM is robust at least to some experimental perturbations. However, in general, for ANAPALM and related methods to work, one requires a strong inherent redundancy of the structures. For random distributions of molecules, this technique is not going to work at all. And there has to be a certain level of consistency between the training data and the testing data. Otherwise, artifacts can be expected. But this error map that we proposed is at least one way to safeguard against some of these artifacts. Very importantly, in order to use these techniques, it's very important for anyone to be able to retrain it on other data and not just to use the already trained neural networks. Here, I just want to advertise this platform called ImJoy that was developed by Wei Yang in my lab that is in fact able to facilitate the deployment of a multitude of deep learning techniques, including ANAPALM. And one of the main advantages is that it allows users to retrain these methods on their own data. Just want to finish by thanking again Wei Yang, who is behind both ANAPALM and ImJoy. ANAPALM was developed also with help from Michael, Andrei, and Shen for the experimental part, and ImJoy was developed by Wei with help from Florian Müller in the lab, and also some help from Martin Jelmare and Emma Lundberg. And thank you for your attention.
Welcome everyone back to the second part of Advancing Artificial Intelligence in Microscopy, where we're going to speak about image restoration and nuclei segmentation, so two applications that pertain to microscopy. And when we talk about microscopy images, these are the type of images that we're now going to see a lot and that I will speak about mostly as it's the data that we're going to apply these methods to. So foremost, these are 2D images of fluorescence microscopy or from histopathology, where we have RGB images that typically are coming from whole slide scanners and are 2D, but it can be as well images from bright field modality, in this case, stem cells moving in a microfluidic device, so 2D plus time, so 2D images that change over time. But at the same time, this can be 3D images, in this case acquired with light sheet microscopies. So in the first subtask, we're going to speak about image restoration. And in the overview that Christoph gave you, you already seen a lot of cases where you have a certain task that demands an input image. And this is a case where the input image is a continuous valid image, and the output is again an image, in this case, the restored image. And why is this important? Image restoration in total. This is important in microscopy specifically because you always are kind of constrained by the trade-offs of your sample health, specifically in live cell imaging. So you always have to follow certain trade-offs in terms of your photon budget, your light exposure, speed, spatial resolution, and all of those three things, for instance, cannot be attained with a maximal resolution. You always have to trade one for the other, and this results in your images being almost always less than from what you can perfectly attain in a single time. And to give you a practical example of that, this is an imaging of a planarian flatworm that people did. And what they wanted to do is track those individual cells at a certain light exposure that does the worm, or where the worm is still happy. And I just go back again. And this is the quality with which they can maintain that. And of course, you cannot track those cells with this image quality. And machine learning restoration allows to restore an image that is acquired under these conditions, but with an image quality that you cannot attain with the laser setting that is depicted here. And how do we do that? In this concrete example, what we did is we fixed the worm. So we fixed some examples of those flatworms and imaged them at different laser exposures. So we had a low SNR setting or different low SNR settings and a high SNR setting here to the right. And we imaged the same worm with different settings, so the same structures with different settings, and thereby could generate a lot of volumetric acquisitions that are, or image pairs of low SNR and high SNR images that are pixel-wise, pixel-perfect aligned, but one corresponding, the left in this case corresponding to the low SNR condition, and the right one corresponding to the high SNR condition. And then we just applied the trick that Christoph, the general training scheme that Christoph already mentioned in the first part, and gave you ample examples of, where we just trained a regression model, in this case a deep convolutional neural network model, with those image pairs. So we had on the left side, we had the input image pairs that are consisting of a lot of cropped volumetric stacks of the low SNR condition, and to the right we gave training examples or as training targets. To the further right, the high SNR ground truth stacks that are corresponding to the low SNR targets. And at the beginning, and we trained the network, or we adjusted the parameters of the networks to give us the best restoration possible compared to the high SNR ground truth. And as you can see at the beginning, the network output is a very blurry instance, or just a very bad job at actually cleaning those images. But the more training progresses and and the parameters of the networks are getting optimized to minimize the difference to the highest in our training target, the better this output becomes. And of course, when you do this for your image restoration task to clean up your images or you denoise your images, an important aspect is always how to validate your results. So whenever you do that, you have to set aside certain stacks that the network was not trained on, and then apply this machine learning based or deep learning based pipeline for denoising on those stacks. So this is a very important component of that. If you now do that on stacks that the network hasn't seen yet, and this is lowest in our complete stitched volume, where you can readily apply such networks because they're so fast, even on large volumetric samples. You see that even on unseen stacks, the network was not trained on. The network restoration is pretty good. This is of course only a visual comparison, but if you do that now and overlay the restoration with the actual ground truth that in this case you still can acquire because this is a fixed worm, you see that most of those restored, or actually all of them, all of the nuclei that were restored are actually part of the ground truth. Another way of validating your image restoration results is comparing them with classical baselines methods. So for instance, a low pass filter or a median filter. And I always would recommend doing this to see how well even a classical method would perform on your data. One of the fundamental core principles of machine learning is always that your training data distribution is identical or is reflected in your test data distribution. So when you go far away and apply any trained network on data it was trained for, it will break in very surprising ways. And to give you an example of that in the context of restoration, here is a restoration denoising network that was trained purely on 2D images of nuclei, with to the left the input and to the right the ground truth image pairs. So a very noisy images of nuclei. And at the same time, a different network was trained on flywing data, so a totally different biological structure. So instead of nuclei, blobby-like structures, we have membrane-like structures. And we trained two networks that are independently were trained on the respective training data. And so you get a nuclei restoration model and a flywing restoration model. And of course, for both models will on their respective training data and test data. So their data that was nuclei-like for the nuclei model and data that is membrane-like for the flywing model, it does a pretty amazing job to restore those images. But of course, now if you apply a nuclei or the model trained on nuclei data on the flywing data and vice versa, the results will be quite horrible. And this is what you see. So if you apply a flywing model that only has seen membrane-like training data during training, and apply it to nuclei images. So on the left side, on those noisy images, it will actually denoise the background quite well because the background is comparable in both cases. But the actual structures of the nuclei, it will result in little membranes appearing in the outside, and vice versa. Nuclei model applied on the flywing data, it will make everything blobby-like. So this is a very important aspect that you should not do. And this, of course, is kind of the worst case scenario because those two data distributions were so different. But this is just a reminder that the more stereotypical your training data is, the more the CNN can actually leverage the specifics of your training data, but the easier it is to fail on different data techniques. So this is one thing that you always have to make sure that you not deviate from that. And this is particularly important if you would use any network from the internet. And so there's a danger inherent to that strategy. So it's always a way to know what is applied on any level of the data. And to just give you a short outline, currently I described to you an image restoration method that uses pairs of low SNR, high SNR, or it works for different modalities too, but you always had to have pairs of the input and the output. And this is, of course, sometimes very cumbersome to do in microscopy, where you cannot arbitrarily acquire input and output pairs. And there are recently, in the last 2 years, a lot of methods for denoising that get away with this constraint that is very hard in microscopy sometimes to attain. One of them is noise to noise, one of them is the other one is noise to void or noise to self. And Florian will speak about that in depth in part 3, but this is just a short outline and last slide for the denoising part. So these are new methods that Florian will speak in part 3 about. In the second task, we're going to speak about image segmentation. And here again, the input is a continuous valued input image, and the output now is again an image, but this time every pixel has discrete values. So only a certain amount of classes, for instance, background or foreground. What we came up with, or what we thought of going to solve this problem for this challenging task, is instead of predicting bounding boxes, do something a little bit more elaborate that is in line with the cell shapes that we see. So instead of bounding boxes, what we predict instead is predicting a radial star-convex polygon for every of those pixels, which is more shaped, which is a better representation of the shapes that you can find in normal nuclei found in microscopy images. And in practice, what we do is for each of those pixels, we predict two things. First, a so-called object probability, which is the probability of being the center of an object. And the second part is we predict for every of those pixels radial distances or along a certain set of predefined rays. And this can be 16 or 32 predefined rays in this case. And this gives for every pixel of the image, this gives you a shape representation, a polygon that gets or is a better shape representation for the nuclei than, for instance, a bounding box would be. And these